Hi, everyone, and welcome to the 16th Byte-Size Talk. I'd like to start by thanking the Chan Zuckerberg Initiative for funding this series of talks, and to you, the NF Code community, for receiving them so well. If you'd like something specific presented in the series that we haven't yet covered, or you'd like more details on something that we have, please reach out to us and let us know, and we try to make that happen. So today's talk will focus on how to use existing data and add new test data to NF Core modules. And we have Kevin Menden from Cubic at the University of Tübingen in Germany presenting. If you have any questions for Kevin during the talk today, please type them out in the chat on Zoom or on YouTube, and I will read them out at the end of the talk. And you can also continue the discussion on the Byte-Size channel on Slack. So thanks very much for joining us today, Kevin. We are looking forward to your talk. Over to you.
Yeah, thanks for the introduction, and hello from my side as well to everybody. Today we'll be talking about modules and NF Core modules and test data for these. And specifically, we will be we will be talking about how to use the test data that we have to create tests for your modules, and then also what to do if you don't find the data that you need for your specific module that you want to add to the NF Core modules repository. So I would like to start with a quick recap about the tests for NF Core modules. So this is covered in other Byte-Size talks already. And there's also lots of documentation online, so be sure to check that out. But just as a quick reminder here, for every module that we have on NF Core modules repository, we have unit tests for Docker, Singularity, and Conda. And these tests are basically they consist of several little scripts, so to say. And the main script where the test code is located in this directory here, so the NF Core modules repository, and then under the tests and software directories, you will find for every module a separate directory. And in this, there is a main.nf file, which contains the main code. And there's also a test.yaml file, which we'll look at in a second as well. And then there is the pytest software.yaml file in the tests config directory. And in this file contains short entries for every module. So you have to be sure that your tests are in this file, that the tests are actually executed by GitHub Actions. All right, let's have a look at one of those main.nf files. So this is one from the fastqc module as an example. And so it's, as you see, it's a really simple file. It's really just you include the module here, fastqc, from the software directory. And then you define a quick workflow where you basically just do two things. First is to define some input data, which is the thing we will be talking about today. So you see here, you define file, and then you link to the test data parameter. And then you have a couple of keys to define your test file. And we will be looking into this in a second. And then you just really run the module on this input data and get some output. And that's basically the test. And then next thing to do, or next thing that comes with this is the test.yaml file, where you define some information about this test. So in this case, you say, okay, the name of the test is fastqc for single end data. You have to define the command for the test, which is in this line, and a tag for the test, which is used to run the test by the GitHub Actions, GitHub Actions scripts. And then also importantly, we have some the output files defined. So in this case, this module produces both an HTML and a zip file as output. And usually there's also MD5 sums in here so that we can really see that the output of the module is always the same, which is not the case in this case, but it doesn't really matter. All right, so these are basically the files that make up the tests. Now you might be wondering how you actually know these dictionary keys that I showed you earlier. So as a quick recap, to define the file that we want to use for testing, we have this command file, which is an extra command to define a file. And then we have the test data parameter. And within this parameter, which is basically a dictionary, we look for the test1 fastqc gz entry, or entry of this dictionary using the SARS-CoV-2 and Illumina keys. So, and you might be wondering, how do you actually know where to get this data from, and where to get these keys from, and what files exist there? So for this, the most important file is the testdata.config file, which is stored in the tests config and test directory. So this file is basically used to define all these different test data files that are available. And it has a link to a test data directory, which is the NF Core test datasets repository, and specifically the modules branch of this repository, which we use to store all the test data in. And you can see here that there are links to specific files in this directory. And these links are again then linked by the in this dictionary. So this is just a small example of this whole test data parameter dictionary. So the reason we do it this way is basically that we are able to change these links at some point without changing the code in the test main.nf files of the modules, because you will only have these keys here, and you will have these entries defined in your script, and so you don't really have to care about what's happening on this side of the code. So this is where I can look for the keys in this file and look for which data you need. And then you might still be wondering, but how do you actually know where to get this data from? And so for this, you have to have a look at the NF Core test datasets repository, and again, the modules branch of that. So this repository is again where we store all the data sets that we use for testing. And there's a quite large README file on this repository, where we try to explain some information about all of these different test data sets. So make sure to have a look at this README file, and then also just go through the repository and look for the data sets that you need, and make sure that they are the correct ones. And this way, you should really be able to find the data you need for your module. So currently, we have mainly 2 genomes available. The one is from SARS-CoV-2, and the other one is a subset of the human chromosome 22. And the reason for this is that the SARS-CoV-2 genome is really small, and so we can really use the full genome and use all kinds of files for this genome without having large data sets. So this is important for 2 reasons. So first of all, we want to have small data sets for testing so that the whole test data repository doesn't explode. And also the tests are much smaller, of course, if you have small data sets. We also have the human genome, but again, only a subset of the chromosome 22, because otherwise it would be too big. And for this as well, we have just a lot of small little data sets available. Currently, we only have genomics data available on the test dataset repository, and also some not so standard genomics data for some specific tools that have been added by users. But there's quite a lot of data on there already, so you probably will find what you need on there. All right, so how then do you actually use that data when you write a test? So the first thing you have to do is you identify the file you need, as the way I just mentioned. So you go to the test dataset repository, you go through README, you look if the file is available, and then you double check by actually looking where the file is located. And then you use that information to look for the corresponding keys in the test data.config file. And then you can really just use those keys to link the file in your test code. And at that point, you should really be able to just run your test and finish your test code. And then basically you're done. That's all there is to it, basically. Unless, of course, you can't find the data that's necessary for your module. So, of course, we don't have all kinds of data sets online, and there's all kinds of bioinformatics tools available, and some of them may need specific data to run the modules. So it's quite possible that you'll find that your data is not available. So in this case, it's not a problem. You can just upload the data yourself, and this will also be really helpful for us. So how to go about this? So the first thing is, again, read the instructions in the README on the test dataset repository. We have a few lines in there about how to add new test data. So make sure to check this out. And then next thing is to think about a suitable minimal test data set. So here, the optimal case would be if you can find something that goes with SARS-CoV-2, because this is sort of our favorite genome now, because it's really small and there's lots of data available. So hopefully you can do something with this, because then everything stays small and we don't have to add a new genome, for example. And once you identified something, it's the best to ask in the modules channel if that's actually working or if there's some other opinions about this, just to get the discussion going if that's suitable for the test dataset repository. Once that's okay, you can basically go ahead and start to create a pull request to the test datasets repository, again to the modules branch, and then ask for reviews in the modules channel and try to get your pull request merged. And once that's merged and your data is basically available, then the last thing you have to do is to add an entry to the test data config file, again, in the modules repository. So there you will have to do a separate PR to get that entry merged, but that shouldn't be a problem. And once that's all done, then basically you can use your data just like any other test data that is available with a fresh copy of the modules repository, of course. So just a few words about guidelines for new data, because we want to keep all the data in this test data set repository somehow similar, and we want to keep it also small. And so the first guideline is to make the data as small as possible. So of course, it has to be big enough that some output is actually produced. So some tools only work with a certain data, so it can't be too small, but also the output doesn't have to be meaningful. It's enough if something is produced, and just try to keep it as small as possible so the whole repository doesn't explode. And of course, try to use existing genomes if it's at all possible. And then also, if you add new data, try to adapt your data to these genomes. So for example, for the human genome, if you want to add human data, you actually have to make sure that there's a certain region of the chromosome 22 that we use, and you should make sure that it's using actually that region, your data that you're uploading, if that's suitable here. And so try to make it really adapted to the genomes. And it might be that it's not possible with these two genomes. Then again, we will discuss this on Slack, and then maybe we add a new genome if that's necessary. And then also, we really like standardized names here on the test dataset repository. So you might have noticed that the files are called test1 FASTQ, for example. And the goal is here to have the files similar across different genomes, so that we basically could, in theory, just swap the genomes and the test names would be the same. So here we really try to, as much as possible, standardize the names. And then a few more words about adding test data, because we know that it can be quite a lot of work and to add new data, because you have to go look for data. You might have to produce it. You might have to, cut the certain regions that it fits to the human genome, for example, and you have to verify that it's the same genome for COVID, and so on and so forth. So it can take quite a bit of time, but that's totally worth it, because it will not only help you to actually get your module on the NF Core modules repository, where it can be used by lots of other people, but also the test data might be used by other modules in the future. So it's really helpful, not only for you, but it's helpful for all of the community. And we really appreciate this. And then in the future, it might also be beneficial for other communities, because the goal is, of course, at some point, or the dream is to merge different test data set repositories from other initiatives and from other communities, and to have one test data repository where all this data resides in the future. And yeah, so if you add some data, it really helps, I think, a lot of people in the bioinformatics community. So even if it takes some time, it's really appreciated, and it will help all of us. So with this, I'm already finished with the talk about this small little issue about modules test data. So if you have any kinds of questions, please go ahead in the chat or ask directly.
Thank you, Kevin. Yes, as Kevin mentioned, if there are any questions in the chat, please put them in the chat, and I will read them out. I'll wait for like a couple of seconds to see if anything pops up. Right, there doesn't seem to be anything that's immediately pressing here that's come up, but please feel free to continue the discussion on the Slack channel. And I'd like to also say that we will be sharing the slides and the transcripts for this talk with you on our website, so please stay tuned. And let's thank our speaker again today, Kevin. Thank you very much for taking the time to present. And thank you all. Thank you, everyone. Catch you next week. We will have Edmund Miller presenting byte work.